Okay, I guess thanks for the introduction, and it's a great honor to receive this year's O'Donnell Award for in science, and I will leave the more acknowledgement to tonight's reception. I just go ahead to my talk, and I think it's raining late. People probably already get tired. So I normally prepare a talk like 1 day before presentation, and this time I realized I made a mistake because it's just for the general audience, I have to try to avoid use very specific technical terms, and I end up I have to work till like 2 a.m. this morning to assemble 20 slides. Okay, this is a daunting task for me, and I'll try to explain what we're doing and see what's going on, and to some extent it's good. The last session, we talked about some kind of neurological activities, and the protein where I'm going to talk about, ion channel, actually underline all these neurological activities. So we call this ion channel, no pointer? Okay, well, I guess just, sorry. Oh, there's another one here? Is this one? Which button? Oh, the green one. Okay. Yeah. Okay. More guns. Okay. So, so it's a carrier of thought in a simple way to describe it. So basically, thought is what you take every day, and also one of the sort of you commonly see materials. And also, salt is abundant in your body, so I use sodium, or it's kind of awkward. I have to figure out which one to push. Using sodium and potassium example, those basically is the most common salt you take into your body, and it's also most common salt in the cell. And I use this as an example to describe the function of ion channels. So basically, your cell is surrounded by lipid bilayer, we call membrane, and it's not permeable to charged particle like sodium ions or cations like sodium or potassium. So in the cell membrane, they have two kinds of proteins sitting there to allow the passage of those particular cations. So one is called channel, and the other called transporters. So the difference between transporters and channels is channel is a passive device. They're sitting there and allow the ions to go along the chemical, electrochemical gradient, and they don't need energy. Okay, so it's a passive device. But for transporters, like sodium potassium pump, as an example here, they actually transport the ion against the gradient, which requires the energy. Okay. So, of course, for today's talk, I'm only going to focus on ion channel, not talk about the pump. And one of the key sort of functions of ion channels involving sodium transporting and potassium transporting is this sets the membrane potential. Okay, so basically, for the inside the cell, you have a very high concentration of potassium, but relatively low sodium. But outside cell, you have a high concentration of sodium, but pretty low concentration of potassium, and those sort of the different concentration sets the membrane potential. Your membrane, cell membrane, actually is look like a charged capacitant. It has minus 70 millivolts difference across it. So, and the work of sodium potassium channel actually underlines one of the basic activity in a neurological, like excitable cell, like a nerve cell or muscle. That's called action potentials. And this action potential is also underlying all the neurological activity, like your thinking, movement, memory, everything. Okay, so all this is underlying activity is actually because of the action potential. So I just give you this as an example. Normally, as I mentioned, cell membrane is hyperpolarized, we call it. It has roughly minus 70 millivolts. When it gets perturbed, let's say it's moved up to minus 47, minus 40 millivolts because of some non-specific channel opening or some other activities. Then the sodium channel has opened. And sodium channel open, then high concentration because of high concentration of sodium, they flush into the membrane, into the cell, and this depolarize, makes the inside become positive. And then this triggers the opening of potassium. Okay, then the potassium open, then they will repolarize the membrane. So they basically, this is look at the membrane, the voltage change across the membrane. This is called the action potential. And this action potential actually moves pretty fast along the cell, nerve cells. So once it gets activated, this part get activated, then you see the membrane potential sort of rise up, goes down, and then shift. Then they move along the cell. This is assume it's an axon, so move along at a very fast speed. Okay, so basically all this activity requires the function of channels. So this lab studies the structure and the function of particular group called cation channels. And we normally use two very basic techniques. One is protein crystallography, and that is we sort of mix the protein to arrange in the crystal, single crystal, and then we shine with X-ray. Use X-ray, you got these diffraction spots, nice spots everywhere. And we wanted, and once we have this diffraction, we go through some mathematical back calculation, we can get the three-dimensional arrangement of all the atoms from that particular channel proteins. And the other functional study we use is called electrophysiology. Because it's a channel, it permits charged ion, and that's basically, if you measure it, you will see current. And here you have, let's assume there's some channels inside the membrane, and then we put electrode on both sides. When the channel open with current pass, you will see current. So with charged particle move through, you will see current. So here, example, the channel is closed. Then suddenly one channel opens, you will see a current, and then it close and open. And then here, actually, you see two channels open at the same time. So you see these steps. And from this, we can sort of understand the dynamics of the channel and how this channel is regulated. So we combine these two techniques to understand how the channel functions. So for ion channels, they have two basic properties, and it's very important to define their function. One is so-called selectivity. Okay, so let's assume this colored sphere represents different kind of cations here. And this channel only allows those spheres which represent, those ions represented by green sphere to pass through, while exclude others. Okay, so based on the selectivity, we can classify channel as sodium selective, potassium selective, calcium selective, or non-selective. Some cation channel actually not very selective. The other thing we call it is so-called gating. That is opening and closing of the channel. Your channel, you cannot open all the time. Otherwise, you can cause a lot of problem. And also, you cannot close all the time, right? Then that's also destroy its functions. I mean, one way people sort of, one common things people encounter by, this venomous snake bite is because those venom actually blocks the channel, so that channel can no longer conduct, and causing all this death and other problems, and including some like scorpion venom and the spider venom. They all actually targeting channel and block the channel, make it not open, unable to permit ions. So to open and close, there's two sort of in a very sort of vague term, there's two kinds of stimuli cause the channel to open. One stimuli is the binding of some ligands. Those ligands can be small molecule like calcium or some other ions, or some can be big protein molecules. So we call this a ligand-gated channel. The other big group is channel close and open in response to the membrane potential change. So this sodium and the potassium channel I just mentioned, setting the membrane potentials, those are channel actually are sort of voltage gated because they sensing the membrane potential change and then control its opening and closing. When the time is right, they jump into action. Okay, so the talk will be focused on potassium channel. So I'm not, even though we are also interested in some other cation channels, but just for simplicity, I will focus on potassium channel, which the lab spend a lot of effort on. And so potassium channel are tetrameric. What does that mean? Is basically it has 4 identical components to assemble and create a whole. And it's through ion actually going through that holes, sitting close. So for each subunit, let's say subunit 1, they have a very basic component. We call it has 2 cross membrane spanning plus some other stuff here. So this is a very basic unit. Every potassium channel have to have this part. Okay. So, but on top of that, you can attach some other stuff to this so-called basic component, and this makes so those extra stuff actually provide some regulatory role to that protein. For example, here they have a so-called voltage sensor attached to the protein, making the channel to be voltage dependent. Sometimes they can have a sort of ligand binding domain attached to the protein, makes the channel to sensitive to some specific kind of ligand. Also, some channel can be both ligand gated and voltage gated. So no matter how complex, this is a very basic unit. All these channels, all potassium channels have it, and this is the components which actually create a hole in the middle. So show here is a movie. Can someone just? Yeah. So this is just the structure of a potassium channel. This is a very the prototypic potassium channel. They call it KCSA. Actually, this structure actually guarantees the Nobel Prize for my previous mentor, Ron McKinnon. And they got this structure, and you see this. I colored each individual subunit into different color, but these 4 subunits basically are identical. Okay, and they create a ion permeation passage in the middle. Okay, so next is a cartoon representation. It's the same as the other program. This is not a movie, don't worry. Okay, so I will remind you. So this is, I only show 2 components or 2 subunits. Okay, so side by side, just remove the front one and the back one to to let you see the passageway in the middle. So basically, they have the so-called 2 membrane spanning segment, M1, M2. That's what I draw these 2 sort of cylinders. And what's interesting is I have to point out 2 particular regions which is very important for the channel. One is this region, colored in orange, and this is so-called selectivity filter. So potassium, because of this region, these potassium channels are very selective for potassium and won't allow sodium to pass through. Okay? So this is the region, and when potassium goes through this region, it's in the dehydrated form. Basically, in general, all this cation in solvent, it's in the hydrated form, but when the potassium have to go through this, they have to remove the hydrogen shell and pass through it in the dehydrated form. The other region I would talk about is this so-called bundle crossing, because these 2 so-called helix structure of this protein, they form the bundle crossing and they pinch shut. And actually, this channel is not permeable to ion because this part is closed. We call it closed conformation. So we're gonna touch both selectivity and gating a little bit to let people have a sense about how those channel protein works, particularly the potassium channel. So first I'm going to focus on so-called selectivity, and we're going to zoom in this region, look at the atomic arrangement in this area. So this is a motion, if you can just, yeah. So show here is the so-called selectivity filter. It has, so all this, we colored most of the atoms at the joint or tip into yellow, but only colored those oxygen atoms, which involving ion binding in red. So just to sort of emphasize it. So basically, we see this selectivity filter. They have 4. Those oxygen atoms forms 4 contiguous cage, we call it. and with iron, potassium can bind in the center of the cage. And in the structures, you can also see a hydrated potassium and iron, okay, surrounded by roughly 8 water molecules. And what's interesting is, even though we see 4 potassium inside the filter, at a given time when the channel is conducting, on average, there's only 2 potassium are occupying this filter. And because they are too close, if 4 getting together, the energetic is not going to be stable. The distance between these 2 ions, like 3 angstroms, is just too close for charged atoms to sit in there. So what happened is these 2 ions sort of occupy in the filter at a given time, and they hopping. Okay, hopping in a sort of, we call the 1-3-2-4 configuration, and then go either out and down. I have 10 minutes, that means, okay. I have plenty of time. I will be a good citizen. Okay, and because channel is a passive device, this process can go either direction, depends on the driving force. Okay, after we get to the structures, there's a sort of heated debate about what's the bottom line of selectivity. Why this particular structure give rise to the potassium selectivity for the channel. So from the structures, the structure actually fit to a classical so-called snug fit model. So bottom line is what this suggests is because of this oxygen ligand, they create this cage. The size of the cage is perfect for potassium ion, but sodium actually is smaller. But because it's small, and those oxygen atoms cannot chelate or touch the sodium ion properly, and make dehydrated sodium ion very unhappy, and they prefer to go back to solutions instead of stay in the filter. But here they make it sort of perfect for potassium. But computational biologists, they did all kinds of calculation. They gives many hypotheses, and it makes the field very confusing because those are just some of the hypotheses they raise, including coordination number, chemistry, property of those ligand, and the dynamics, etc. Okay, so then I will read one studies in the lab. We try to address these questions. Why they have this? What is this sort of structure? What's special about these structures? Okay, and the other thing is why they have 4. Right? If it's snug fit, probably 1 is enough. All they need is just 1 potassium ion sitting there, and if it's perfect for potassium but not for sodium, then probably it's good enough for selectivity. So to study this, I have to introduce another channel. So this is a KCSA we have been using for the last couple of slides. Now I'm going to introduce a channel we call MthK channel. This channel is non-selective channel. This can allow sodium and potassium to go through equally well. No difference. They cannot differentiate that. But KCSA potassium is very selective. Okay? So overall architecture sitting in the memory is very similar, but the key difference is the selectivity filter region, which is supposed to be responsible for potassium selectivity. So if we zoom in and look at the filter, so this is a KCSA, a canonical potassium channel filter with 4 cage. But when we have this channel structures, we notice that they only have 2 cages, contiguous cage. And these 2 cage, equivalent to the bottom 2 sides, we label sides 3 and 4 of canonical potassium channel. But this channel is non-selective, even though they maintain 2 sort of equivalent sides, presumably suitable for potassium binding. Then we did some engineering, going from this filter, we did some, I think, pretty clever mutations, and we created a filter which contains 3 contiguous sites. And the channel is still non-selective. And this 3 sites basically has virtually identical coordination chemistry as this site 2, 3, 4 of canonical potassium channel. The channel is still non-selective. Then we also go from here, we create another channel which contains 4 contiguous sites, just like canonical potassium channel. Now, once we hit 4, the channel becomes potassium selective. Very interesting, but we see this phenomenon, we still have no explanation yet. Why once it's hit 4, the channel all of a sudden become potassium selective? And this is still ongoing project, okay? So basically we raise more questions than answer the questions. But anyway, so now I'm going to touch a little bit base about the gating issues. The gating is, in other words, is just the opening and closing of the channel. So this bundle crossing, as I mentioned in this case, it represents the channel in the closed state. So when the channel conducts, this part has to be disrupted. How does it get disrupted? Okay. So what happened is when the channel opens, this so-called pore lining in the helix, which forms bundle crossing here, they have to spray open. So this is a side view with two subunits with opening and closing and opening of channel by bending of this helix at one of the very sort of conserved amino acid. We call it glycine gaining hinge. And if you flip this 90-degree view from an intracellular side, we'll see this opening is just like an opening of a camera shutter. It's just the sort of dilated open. And this creates a big hole, intracellular holes. And this hole basically allows a hydrated big hole, a hydrated potassium to get into the channel, dehydrated, and pass through the filter. Only this filter region which need the ions to be in the dehydrated form. The rest part, you don't need to. So once it's pinched shut, this hydrated potassium ion cannot get in, and basically the channel cannot permeate. Okay, so this is sort of the open. But the pore cannot open by itself. You really have to have external sort of stimuli. So I mentioned briefly in one of the previous slides, there's two kinds of stimuli. One is so-called ligand. So basically, they have this sort of ligand binding domain, part of the protein, attached to the ion conduct permeation pore. And when ligand binds, some conformational change occurs here, leads to the opening. And the other sort of stimuli is voltage, because they have some extra stuff, transmembrane region, we call voltage sensor, sitting in the membrane. When the membrane potential change, something happened in this part and leads to the opening and closing of the channel. So I'm going to use a ligand-gated channel as an example, see how this ligand-gated ligand binding domain regulates the pore opening and closing. So this is a movie. So here is a calcium-gated channel. We use it as an example. Top portion is the membrane spanning portion, the pore, ion conduction pore. Can someone quickly click this? Okay. So they have a huge bottom parts, okay, which sensing the ligand. And in this case, the ligand is calcium. You can see those yellow sphere represent calcium. There's 8 calcium binds to it. And this channel happened to be in the open state. Basically, this is the ligand bond open channel with this gating, this huge ligand binding domain. And those ligand binding domains form the ring structure. So we call it gating ring. Okay. So to understand how this rings regulates the channel opening and closing, we can determine the structure of the rings in ligand bound and ligand free states. So ligand bound is represent open conformation, ligand free represent closed conformation. When immediate sort of visual sort of conclusion is very simple. Actually, in the open states, the gating ring is in a more expanded form. You just look at the distance. Okay, the distance, if we just this is the diameter. In the open form, it's 8 angstroms wider. Okay, so this can give us this is going to be the last slide. Very quick view. What happened? This is just the linear interpolation of how the gating ring open and close. This is also a movie. If you can click this. So this is the expansion. We just look at the ring parts. But if we look at this in the context of the channel, when this gating ring expands, they basically convert the chemical binding energy to a mechanical force. And sort of this is just a simple mechanical device. Expansion of the gating ring drag this pore lining in the helix and cause the pore lining in the helix to bend and then spray open the intracellular gate, allow the channel to open. So this is just so I'm going to stop here and acknowledge the people. Okay, and this is some simplified view. I try to be simplified. If you still get confused, I apologize. View of how the channel works, but there's all kinds of channels. They may do different things. There's still a lot of work need to be done. And so those are the people who worked in the lab, because all the achievement is in the lab depends on those hardworking people. So those are in the early stage of my lab. There's a picture taken in 2005. This is just taken pretty recently, and people change, and you will see people come and go. And of course, I stay. But one bigger change is I don't have much gray hair at that time, but now it's just filled up with gray hair. You can what's the big toe of doing science, I guess. Particularly for the last couple of years, to getting those fundings become a big issue. So all this funding supported the research, which are very important. And I just listed chronologically. UT Southwest Endowed Scholar Setup provides the startup, and I have support from Packard Foundation, Sarah Scholar, Welch Foundation, Magnite Endowment Scholar Award, and NIH, and lastly, Howard Hughes Medical Institute. So thanks for your attention. And I would like to question. So I finish. Okay.
In your electrophysiology experiments, to what extent can you or have you studied the effect of different counterions on potassium selectivity?
Oh, actually, I should emphasize this. So because this natural selection does not need the potassium channel to discriminate against other cations, because the most abundant ions is sodium and potassium. So actually, potassium channel can conduct rubidium, cesium pretty well, which is slightly bigger than potassium in the same, line of this elementary table. So they allow those big ions to go through, but they won't try not to let sodium to go through.
I guess my question was more centered on the effect of the counter anions.
Oh, counter anion? No, they won't allow cation to go. I'm sorry, I saw the other cations. Counter, no. So ion channel most ion channel are pretty specific. If it's cation channel, only cation.
But could some counter anions inhibit the selectivity of a cation channel?
Well, I think the selectivity is pretty electronegative to some extent. So I don't think, let's say, chloride can actually get in there. It's just not going to be a great environment for chloride.